Welcome to the MOOC NPTEL course on Bioengineering and interface with biology and medicine. Today we have reached to the last lecture of this course. In last several lectures, we have discussed about various biological processes associated with cell cycle, development, cloning, cell reprogramming. We have also discussed many other interesting biological concepts in genetics and cell biology. We have seen how DNA tools and biotechnology is driven by lot of technological advancements, different techniques we have talked. We have also discussed about proteins and protein related technologies. In addition to these topics, I think it is very important for me to emphasize that there is a need to have ethics in research and publications. So, today we are going to talk about this topic, which is not directly linked to the biology, but it is highly relevant when you are doing science and research. These kind of discussions, these points are very important for you to remember. So, let us start today about ethics in research and publication. So, whether you make engineering devices, products or scientific basic research, ethics in research and publication is of paramount importance. Remember the story of animal cloning? We talked about how the rise and fall of stars happened in the research of cell reprogramming and cloning, and you have seen also the various consequences of scientific misconduct. So, today let us analyze the reasons for scientific papers retractions. Let me take you to some examples and possible reasons that why scientists do perform these scientific misconduct. So, I have shown you here some of the examples of scientific paper retraction. You can see this paper from Nature Cell Biology. I have shown you another example here where another paper was retracted, one more example here, and in this case even the co-authors were not aware of what were the issues happening, and one of the co-author had mentioned that I was so shocked. In fact, I had absolutely no knowledge whatsoever on the actions taken by the corresponding author. So, I think it is very important that if you are part of a team, the corresponding author should inform all the co-authors about any development which are happening related to your scientific work and the publications. Let us now analyze and discuss what are the possible reasons for retraction of scientific papers. There are increasing number of scientific paper retractions happening, which is really bad and alarming news. If you do the PubMed search, in Jan 2018, they identified almost 9,800 articles were retracted. The report from the Office of Research Integrity and other published resources and additional retraction announcements are also happening in scientific journals, which are also worth analyzing. In journal PNAS, they try to analyze what are the reasons for the scientific misconduct and why scientific papers are retracted, as you can see in this slide in the published article from PNAS. They try to analyze various reasons for possible retractions and found that while a small number of papers, almost 20%, where because of some errors which has happened in the process, people have not made the knowing mistakes, but there are almost 67% scientific papers were retracted because of fraudulent cases, could be suspected fraud, the duplicated publications or even plagiarism. It seems the image manipulation is one of the favorite area for the fraudulent researchers to manipulate and then do the changes in the results. I have shown you here a couple of examples. Let us look at here this image manipulation, where a protein analysis is happening and the blot is actually manipulated. The shown here from the Journal of Cell Biology paper, on the left side the original image, and on the right side you can see the manipulated image, how the bands have been removed to look very different. So, image manipulation by adjusting brightness and contrast is another area where lot of manipulations happen. As you can see on the left side, the original image, and right side the image which is manipulated, just by changing the contrast, how different these bands are looking like. In the next slide, you can see again by changing the brightness and contrast, even the gel which has many contaminants could be avoided, and just a single band is shown on the fourth panel in the right hand side in the image. Again you can see another example where original image was manipulated, and now all the bands which are visible in the lower side, they have been removed, and now you can only see single band from the gel. Looking at the cell morphology, cell under microscopy, again lot of manipulations happen. For example, enhancement of a specific area is shown here in the image which is manipulated on the right hand side. And now you can see that some of the misrepresentation of microscopic images, something shown on the top panel is an image which is manipulated, and now later on if you change the contrast, you can see how this was manipulated. So, general science they looked into the image manipulation very seriously, and their data and analysis showed that large number of publications have actually been retracted because of the manipulation in the images, and therefore, now many of these journals, they have their editors, and they have the the technical team who also looks at the images quite seriously even before the manuscripts are being sent for the peer review process. So, the question is that if you are a PI or the head of a lab, and you do not have very much experience about how to operate these photo imaging softwares or the photo editing softwares, you may not be able to really make sense that what is exactly the correct image or what could be the manipulated image. So, one of the researcher actually mentions here in one of the blogs that my personal experience says that research supervisors and guides, they also need to be trained to detect the manipulated pictures. Sometime because the students can make these changes and manipulate this, and even they may not be able to figure it out. So, it is really important that on one hand one should not make any of these kind of changes, but there should be a stringent peer review process involved, where you can correct these kind of any suspected practices. So, what is acceptable practice for presenting your data and the images? So, the acceptable practice is to show your figure effectively. Of course, that is accepted. You do not want to show just the raw image without any the cropping the background. However, if you are doing lot of color adjustments or color balance, you should mention that in the figure legend that you have used these settings to change or modify this image. And of course, you can put the raw image original figure in the supplementary, which is part of the manuscript, and a reader, when they are reading it, they can actually try to go back and look at the original image as well. So, what is research misconduct? The research misconduct is defined as fabrication, falsification or plagiarism in performing research or reporting research results. The National Science Foundation has defined these guidelines for defining the research misconduct. So, fabrication is making up results and recording or reporting them. Falsification is manipulating research materials, equipment or processes, or changing or omitting data or results such that the result is not accurately represented in research record. Plagiarism is appropriation of another person's idea, processes, results or words without giving them appropriate credit. Source for these definitions are used from National Science Foundation. So, what are the major types of scientific misconduct? One of them is publishing misconduct. Your data is okay, but one has performed unethical practices. For example, to do a clinical study, the clinical trials, the proper consents were not used, and there has been misuse of the samples which are obtained. So, that is unethical practice, which is a publishing misconduct. The research misconduct is that your data itself is fraudulent, the data was generated in wrong manner, which is much more serious issue. Of course, any kind of misconduct is serious for that matter, but changing data or representing in a different form is of course much more serious. And there has been lot of analysis in which way people have done misconducts from different parts of the world. And if you can see here in this graph from this paper, where it shows that large number of fraud or suspected fraud papers actually came from USA. There are many countries part of it, including India. Of course, USA is shown in the largest fraction, not only because there are a lot of misconduct is happening, but mainly because there are large number of publications are coming from USA, lot of research is happening, and therefore, is a fraction of that is also suspected frauds. People have found even plagiarism and duplicate publication in all of these categories from almost all part of the world are actually involved in this kind of stuff, which is not happening on any specific continent. And if you look at the journals where the most of these articles have been retracted, it is alarming to see that the most prestigious journals including Science, PNAS, Nature, JBC, all of these journals they have reported many articles which were retracted. People have also tried to analyze that what is the mean time to retraction by the category, that how many years it usually takes for the fraud to be figured out and the articles to get retracted. But the main question is that what is the probable cause of research misconduct? Is this the career pressure or looking at more sort of funding opportunities? Sometimes there is a hunger for reputation. It is very easy to fake than make. Many times that when you are part of a team which is let us say as a student, when you are competing for some competitions, you are going to the international competitions and you are part of a team which is making some devices, some sort of robots, and then at that time to be the first, to be the top in the world, I think if you change your mind sometime and kind of get engaged into any kind of misconduct, you are using somebody's already original idea and trying to represent as your own, I think that will probably lead to the misconduct, which is sometime that kind of pressure of whether it is career pressure or competition pressure, I think should not affect your originality, which is really crucial. Let us say you are working in a research lab. Now, you are as undergraduate students, you are going to work in a scientific laboratory. Of course, you are most junior over there, and there are many senior PhD students and the postdocs. They are all working in that lab. And you actually somehow get sense that something suspicious is happening, some sort of scientific misconduct is happening in this lab. So, what should be your role? I think because you are very junior that time and you may not know exactly that what should be the best ethical practices for doing research. It is important that you can talk to your senior that you feel that something the way it is being done in this particular result which you are showing is not correct, and this is my understanding, please correct me if I am wrong. And let us say if sometime you are a senior and a junior student comes to the lab, and now you are finding that the junior is doing some mistakes and he is doing some sort of suspicious things and changing the data, changing the gel images and doing something which is just for the publication's sake, then I think it is your responsibility to inform your seniors, to inform your PI to the professor that something suspicious is happening, and let them kind of decide what to be done. Let us think about extreme situation that your professor or the PI itself is actually engaged in some sort of plagiarism or these kind of suspicious activity of scientific misconduct. That is very difficult situation, because you as a student will actually hesitate to say that how to handle this kind of situation. But I think it is still your responsibility to go back and either talk to the professor, but if you do not have courage to do that, you can actually inform your head of the department or the deans, so that they can do investigation. Next question is what are the possible consequences of scientific misconduct? I am sure in the context of discussion of the animal cloning, you have seen that many of the researchers who were involved in doing the fraudulent work, their career got totally damaged, they lost employments, and these kind of retractions are very detrimental for the any researcher's career, because they are always published, they are always out that and the reasons are mentioned that what are the possible misconduct reason in that paper. Sometime authors could be even banned for publishing for limited time, You have seen that in case of the the cloning story, there were a lot of financial penalty were given. In fact, even imprisonment was also given. So, a lot of the consequences you will face if you do the scientific misconduct. So, it is really important that make sure that you are spending enough time to obtain the good results, and you are not kind of in rush doing the scientific misconduct. So, I must say that it is a very nice statement made by National Academy of Science in 1995, that someone who has witnessed the misconduct has an unmistaken obligation to act. So, if you see that some misconduct is happening around you, you are part of a team which is going to compete in the international game, and you feel that one of your colleague is actually doing some sort of misconduct, and if you do not report that just because your friend is involved in doing that, that is not correct, because you have to report that kind of misconduct if you are part of that team, because every co-author or every colleague of that particular work is going to be equally involved in that kind of misconduct. So, it is really crucial for you to take act on this. I would like to acknowledge for this lecture two of my colleagues from Department of Bioscience, Bioengineering, Professor Bhomik and Konda Begel, who has provided me some very stimulating discussions and good references for this kind of scientific misconduct based various articles published in the last couple of years time. So, students, I hope this course has now made you very much excited to study biology. Let us now review what we have learned during these last 8 weeks. During the very first week, you were introduced to the major life problems and how we require interdisciplinary skills to find effective solutions to these problems. We provided you examples of bio-inspired engineering, where designs and models based on lessons from biology have been developed to solve very complex human problems. We then briefly studied about the cell and its properties. A clinician, Dr. Ali Asghar Moyadi, was introduced, who talked to you about brain tumors and how there is a need for engineering interventions for the medicine. During the week 2, you learnt about central dogma of biology and refreshed about the concepts of nucleic acids. We also learnt about the simple yet very crucial lab techniques like agarose electrophoresis and polymerase chain reaction. We further learnt about gene cloning, both theoretically as well as the lab demonstration sessions, where you have seen how to do the cloning, how to perform these molecular biology experiments in a laboratory settings. In week 3, we discussed about several biotechnology tools that are routinely used for addressing many biological problems. These tools included microarrays, RT-PCR and sequencing technologies. We learned how DNA sequencing has revolutionized the field of modern molecular biology and paved the way for next generation sequencing. Furthermore, we learned how these DNA tools are being used for many applications such as food production as well as in plant biotechnology. We also got clinical insights from Dr. Nikhil Phadke, one of the clinicians who uses these tools for patient care to provide better diagnostic and prognostic information to individuals with genetic defects, cancer and infectious diseases. During the 4th week, we dedicated our time for Mendel's experiment, especially pea plants. We discussed the 2 Mendelian laws of genetics, the real life examples of Mendelian genetics as well as cases where there is a deviation from the Mendelian genetics. In the last lecture of the 4th week, we introduced you to Dr. Jayanti Shastri, a microbiologist who provided her perspective on needs of a clinician. She emphasized on the needs for the point of care test, which could help to diagnose a disease at the bedside. Next, in the 5th week, we continued discussing about Mendel's observation and we moved on to Morgan's elegant experiment on genetics. We learned how Mendelian inheritance has its physical basis in the behavior of chromosomes. We further studied the molecular basis of inheritance and learned that DNA is the genetic material through various experiments. You were then briefly introduced to basics of bacteria and viruses. You learned how to perform experiments to distinguish the gram positive and gram negative bacteria. This week was concluded with another motivating session from a clinician, Dr. Kunal Sehgal, who heads the Sehgal Pathology Labs. The week 6 familiarized you with the basic concepts of cell cycle and key points where the cell might lose the control and often cause disease like cancer. We also learnt about several stages of an embryo and other key concepts which are related to the development of an organism. We also learnt the concepts of animal cloning, and then the lessons on evolution was given to you to appreciate the diversity of the life and also gave you the scope to explore the field of population genetics. Furthermore, this week we had Dr. Mala Kaneria from Nair Hospital, who again gave you a very stimulating talk on need of engineers to work towards technology solutions that can help doctors like her. Next week, which was week 7, we started talking about basics of amino acids and proteins. You were introduced to several protein separation technologies such as SDS page, two-dimensional electrophoresis and chromatography. You also had a hands-on session on bioinformatics, where you learned how to study genes, proteins and evolutionary relationships. From this week, then we moved to the last week, the week 8th, where we had major focus on protein separation technologies and proteomic technologies. So, in this week, we performed few experiments which gave you a good understanding that how advancements in protein technologies are now able to provide us a comprehensive overview of various complex physiological processes. We learned how protein microarrays and label-free technologies like surface plasmon resonance can aid our understanding for studying protein interaction networks and finding their kinetics to provide us better understanding. So, the aim of this course was to encourage you all to see biology in a very different way. We hope now you agree that biology is much more than just a subject. There is a lot of scope for biology and engineering to coexist, and bioengineering can offer many unique solutions that can be useful for agriculture, medicine, health and many other applications. Thus, I hope you are now motivated to learn more about these tools and the areas of bioengineering. Before I finish, let me thank the NPTEL head and technical team for all the support in bringing this course to the students on time. I would also like to acknowledge all the course DAs for their very high quality work. Many thanks to my students of IIT Bombay BB101 course, as they keep motivating me to teach biology course to engineers with their very naive but very interesting questions. Also, a note of gratitude to the members of my proteomics lab who have enhanced the course by their lab demonstrations. We need you and your skills to tackle many research problems. Let us continue this journey together.